Hi, I'm going to go over the effects of soil contamination lab and help you set this up. Here in the Power School are some notes that we will be talking about in a moment, but you're going to be designing your own experiment and testing the effect of one substance. So you're going to choose oil, salt, trash, detergent, or vinegar to represent different types of pollution. You're just going to pick one of those substances and you're going to test it on the growth of grass seeds. Now, if you have other plant seeds, that's fine. And you are going to be testing not just oil or no oil, but you'll be testing different concentrations of that oil. So here are some examples of how you can measure out those different concentrations, and I'll show you in a moment more of what that means. Okay, so here's the lab. I'm going to go ahead and open that up, and I'm also going to open up our scientific method notes so that you are comfortable with some of the things that the lab is asking you to write out. So if we look at these scientific method notes, hopefully you know a little bit about the scientific process and how to make a good experiment. But when you do an experiment, here is a very simplified version of an experiment. Let's say I wanted to test whether Miracle-Gro helps plants grow taller. So I would have one group. Now this shows just one plant, but I would never just do one plant. I would do a group of plants. One group of plants would get the Miracle-Gro, and ideally I would do different concentrations of the Miracle-Gro, and then the other group would be our control, and that would be the plant that doesn't get any Miracle-Gro, so that I could compare the one that got the treatment versus one that didn't get the treatment, so I can see, did it really help it grow or not. The control gives it some sort of comparison. And so when you do your experiment, you definitely want to control where you're going to have some plants without the pollution. So one of the things that you need to distinguish in your lab report is what the independent variable and the dependent variable are. So the independent variable is, what do you do to the experimental group? What are you changing? And for this experiment, we are adding the Miracle-Gro fertilizer. For your experiment, your independent variable might be the vinegar or the vegetable oil, the salt or the trash or the detergent. Those are all independent variables, and you're just going to pick one. The dependent variable is what happens as a result of that treatment. So what are we measuring that the independent variable is supposed to affect? So for this experiment, we're measuring the height of the plants. For your experiment, you're going to be measuring also maybe the number of seeds that sprouted, the height, the physical appearance. If you have pH strips, you could do the pH of the soil. But mostly it's going to be kind of measurements of your plants that grow. That's going to be your dependent variable. And you're going to be collecting data. It could be heights of the plants. It could be also adding some data about their appearance, because if they don't look so good, then you want to note that in your data as well. And then you're going to graph your data. So that's basically how a scientific experiment is supposed to be carried out. So let's take a look at this one that you are going to do. So here the introduction talks about some of the contaminants that get into the soil and what causes them. And we're going to look and see how that might affect the growth of our plants or our grass seeds. So you're going to go ahead and put down what the purpose of this is, what supplies you have, your variables, and your controlled variables. In other words, what did you keep the same between your two groups? So even though this one got Miracle-Gro and this one didn't, there were things that everything else should be the same. They should have the same amount of water, sunlight, soil, space, anything else that you can, same number of seeds. So that all should be constant. There should really just be one thing different, and that is the independent variable. Okay, and then here you're going to tell me how you set up your experiment step by step so that someone could actually follow your steps, your procedure, and do the same experiment you are doing. Okay, you can draw a picture, and you're going to tell me how you're going to make your observations. What will you be measuring? Number of seedlings, the height of the grass, the appearance of the grass, the color, that kind of thing. And then you're going to create a data table to record that. And I would like you to make a hypothesis. What do you think is going to happen to your plants? Okay, so let me show you a possible way to set it up. I happen to use these cat grass, I guess it's catnip grass or whatever. It's just a cheap pack of grass seeds. It's interesting, it says grass wheat, so I don't know. And when I look at the back, it says it takes about 4 to 7 days to germinate, meaning to kind of sprout out of these seeds. And it gives me some directions on how to sow the plants. And what I would do is, just to keep it simple, although you can definitely do more, I have 4 cups, and actually you want to have at least 3 different concentrations. So this is going to be my control right here. I don't know why it's kind of looking like my arrow is pointing. Okay, so this is going to be my control. And what I did was I put some damp soil in, and then I have 10 seeds in there, and I'm going to just gently press them so they're slightly under the soil. We don't want to bury them too deep because then this particular seed is not supposed to be buried. It says that you just sort of press them into the soil. And what I'm going to do is I'm going to take some plastic wrap and cover it. But this one, it has 10 seeds. And depending on the size of your cup, you might want to put more or less. And I have, a certain amount of soil, and I'm just going to have water. I'm just going to water it but nothing else. Okay. This one is going to be, I'm going to pretend that I'm doing the vinegar one to simulate acid rain. Okay, so this is going to be, now if you look at our Power School notes, if I'm doing vinegar, then one suggestion is to do 3 different concentrations: 5 milliliters of vinegar, 10 milliliters of vinegar, and 15 milliliters, as well as whatever water you're adding. So as I water, I'm gonna add this vinegar. That's kind of a lot, but I can kind of decide how much I want to do. Okay, we're only doing this for 2 weeks, so it's not like these are going to get really big and have to go anywhere. Okay, so this is going to be, I'm going to call this control plant A, because when I do my data table, that might make it easier. This is going to be B, and I'm going to do 5 milliliters of vinegar. Okay, and everyone's doing their own experiment. If you want to do the vinegar one, you can do that. This one is going to be my C, and I'm going to do 10 milliliters of vinegar. Okay, everything else is going to be the same. It's also going to have 10 seeds, same amount of soil, same amount of water. Okay, and then this one, my paper ran out, so this one is going to be D, and this is going to be 15 mLs of vinegar. Okay, vinegar is representing acid, and this is also going to have 10 seeds. Okay. It would be nice if you want, you could even do 2 cups that are your controls, 2 cups that are your B, 2 for your C, but I just wanted to make it simple under this video for you to be able to see clearly. And so I'm going to go ahead and press these all in a little bit more, make sure I have water, and then I'm going to add my vinegar, and then I'm going to cover them. So if I don't know how much 5 milliliters of vinegar is, if you look on the notes, 5 milliliters is the same as 1 teaspoon. Okay, and same thing if you're doing the salt and you don't have a scale to measure out 1 gram. 1 teaspoon is about 6 grams, so you can sort of figure that out. It might not be as accurate as saying 1 gram, but you might be able to do like 1/4 of a teaspoon, or half a teaspoon and 3/4 of a teaspoon, or something like that. So you have 3 different concentrations. Okay, so I'm going to pause this for a second and go get my saran wrap. Okay, so when I'm ready to, after I've watered my plants and I'm ready to go ahead and add my vinegar. Okay. Ooh, this seems like a lot. Okay. Ugh. Sorry, plants. Okay, so this one got 5 milliliters. This one's going to get 10. Oh, it's not going to be a good one. And then this one's going to get 15. I've already added the same amount of water to all of them, and so, oh, kind of stinky. And then now, to help them germinate a little faster, I'm just going to cover them with plastic wrap, and I'm going to put them in a slightly warmer place in the home. Okay, they don't need sunlight until they've sprouted, because it's really, they don't do anything with sunlight until they have leaves, because leaves are what do the photosynthesis. Right now they have food that's stored in the seed. Okay, so I'm noticing I didn't even label my cups, but I will definitely label my cups, and I will put these in a warm place and see. Once they start germinating, I'll start recording my data, but I might go ahead and just, I might go ahead and still start recording data. Sorry, that's my dog Charlie. Okay, so I might have something like this, where I'll have okay, this will go on for a while, but I might have plant A, B, C, D, and then I can have, I don't know if I'll put anything for day 1, but maybe once it starts germinating, let's say day 5, and then I'll go ahead and continue from there. And we're going to go for 14 days at least, because we want this experiment to be 2 weeks. And I might be recording number of sprouts, the color, the height, things like that. I might say, 3 out of the 10 sprouted. You'll just sort of have to see what sort of information you see that you need to write down. Okay? So again, it doesn't have to be the vinegar one. It can be any of those substances. And I hope you have fun doing this experiment. If you have any questions or concerns, send me an email.